He comes at the end.
He's the lead. He's the lead off speaker.
No. No.
Okay, let me just copy his stuff over here.
He's not.
All right. Let's have our seats. And I don't think I need to introduce Dave Parks, who's going to chair this next session. I think we're looking at this as a kind of an open lab meeting. So in fact, if anyone has slides in their back pocket.
Can I introduce this or okay, everyone's trying to figure out what to say because I conceived of this monstrosity. But those of you who've been in the lab, which is most of you, know the lab meetings and what they're like, which is that somebody puts up slides and everybody asks questions, criticizes, and brings up relevant subjects. So if you have a slide that would be relevant to put up there, you can certainly do it, but the object is actually to interrupt in these talks and to make them as interactive, not just simply to be this. And so our intention was actually to be in rooms like B100 where we could sit around the table.
What's the first slide here?
We got stuck with this room, so you'll have to imagine that you're sitting around the table and look at the other people in the audience.
Right there.
Okay. We'll move
That whole
microphones
folder,
around
it's
if we
got
need to. But
the,
actually, I mean, I think you can all hear my one.
the C one is the, so that's playing, the other one
Okay.
it's Marty's, and it's the first one. This one, let's see.
This one here?
Yeah, it has them in
Dave
it.
Parks does not need any introduction to people from the Herzenberg lab. Dave Parks to anybody else
Make sure you
is
don't run out of
probably
power.
the person who's made the
Okay, yeah, you put it
single
up.
most extensive contributions to flow cytometry technology. He and Wayne Moore, as I said before, have been doing flow work forever. And almost every new thing, David put the first dual laser fax out for anybody to use as a long-term use, and Randy Hardy and Kyoko Hayakawa did their work on that, and everybody else. That fax went to the Smithsonian Museum. It's sitting there. So that's, David is a museum piece. And for that, I have to give you a medal.
Okay, so, all right, so Stephen is going to lead off. Yeah, so kill this.
Oh, okay.
You go. Checking to see if my stuff is coming, is on there. Okay, so, yeah, I guess maybe the thing I should do is give a, since I was, until fairly recently, the director of the Stanford Shared Fax Facility, which was initially explicitly a spin-off of the Herzenberg lab, and has been on in some form or another since the mid-1980s. Give people a little bit of an update on that and say that the Shared Fax Facility continues to thrive and do new stuff. Has several hundred users, it's hard to say at any given time, but has a huge diversity of applications. Most of the people here have worked through a significant array of things, but we've got people doing plankton and protoplasts and a huge array of different applications, which is the function of the facility compared to things like even the Stem Cell Institute, which has more cell sorters at the moment than the Shared Fax Facility, but has a rather limited array of applications that they're focused on. So we have different people who walk in and do something completely different than the person who was on that instrument the last hour. So it's still a challenge. We do a huge amount of training and education this area, and we still do some instrumentation development even. The commercial instrumentation has gotten to the point where it's not easy to just get in there and mess with the boards and or the internal control stuff as we used to do, or to actually build things new. So that has changed. Although we're still, for those old timers, we still have one dyes laser running. We're down to one krypton gas laser, And the old Fax 2 that was originally elaborated out to 11-color fluorescence is still in use in an outlier facility in the next building. So we're still not absolutely out of the things that we got our hands into seriously, and we still get into the current instruments, but we basically have them maintained by the manufacturers now, which is simplified things a lot. So we do stuff related to that, related to instrumentation, methods development, both biological and computer and analytical. And I'll say a little bit of that if there's time. And otherwise, start with Stephen Meehan, who is the lead developer on the called the Cytogeny project. And I guess just
Sure.
it's been on a number of the people here were parts of it, so just let him go from there.
All right. Thanks, Dave. So I've been, I'm trying to think of all the wonderful parties at the Herzenberg lab that I've been at, just as a way of gauging how long I've been here. And I think the first party like this was the 50th wedding anniversary party, and I guess that was in 2004. And since then, we've had great parties for Lee's birthday, for the lab's anniversary, and now for Len's birthday. So these are always sort of milestone events, not just in getting to see the great history of the lab, which I only became associated with in the last 10 years, but also just to remember where were we at with this program at that time. And we've shown that program at each of these events, and it has evolved most dramatically since the last event, the 50th lab anniversary. And one of the things that was the stimulus for this was we had built this package primarily as an experiment planner and a staining panel or stain set feasibility guidance or genie. And that's what we had done up to that point of that last anniversary. And it was at that time that this word at the top here, autocolor, became coined. It was at that last anniversary party. So I'm wondering what major understanding we'll get from your feedback here today. That will be the next word. But autocolor became the word starting in November 2009. And guess what it was that provoked that word? Does anybody have any idea why it suddenly became urgent for us to look at the complete explicit removal of fluorescence concerns from flow experiment planning? Oops, I'm going away from the mic. There was another discussion of a new technology at that time too. And it was no color. It's called CyTOF. And when we were looking at CyTOF, we realized that of all the great things it was giving, it was nice, but we realized that, yeah, we really have to focus harder on the issue of taking away the fluorescence so that these faster sorter technologies can respond to the fact that this is a primary concern. So Lee and Len really zeroed in on this issue, both with what I'm doing as well as with what David will talk about later in the area of auto compensation. So we have auto color and we have auto compensation, and then just this last month, Len got another automobile. So, and it's a nice, nice black Mercedes. He'll take you for a drive if you wish. So here's auto color. Auto color proposes just to allow you to come in as a new user, first time experience on the software, no pre-setup required, no issues of cutting the software over to your lab and bringing in your reagent supplies are required. All you really need to do to use autocolor is tell us what the instrument is, which it sometimes is a bit of a challenge. But autocolor basically starts with tell me your instrument. I must know the essentially must know in particular, I must know the wavelengths of the filters. I must know the laser wavelengths. And for more subtle configurations also, sometimes the dichroic mirrors. Is that how you say it, dichroic? I spell it, but I don't often say it. And sometimes those are necessary too. It's usually a reasonable short experience if the facility manager is on hand to supply that information. Then the biologist can quickly go into the autocolor workflow, which is tell me about your markers. In this case here, they're just going to enter in a number of T-cell markers. And they're basically just going to type in those letters that you see in the red highlighted column. They're going to pick the markers they use in their stain set. They're going to pay no attention to what channel they put it on for that instrument. And it's all a fairly quick, that short little exercise there could be up to 2 or 3 minutes just to type the markers in. And then they're ready, more or less. They can add a dump channel as well. But at that point, without having pre-configured any reagents, with only supplying the instrument, they are ready to now see the magic of autocolor. So at this point, we've entered in a viability or live dead marker, we've entered in a dump channel, and we've entered in a fairly more advanced high-dimensional protocol. And if we want to lock dyes, we can, but generally, we encourage people to go immediately onwards to letting autocolor do its work. Another thing that they can do, I'm showing it here right now, but they can do it later, is to actually tell us what these markers express on, what cells they express on. And you can enter in more than one cell, as we're showing here. And for each of the markers, they're entering in the information of how much binding they think the antibody will have on the antigen. Again, they could have just entered the 8 markers and gone right to autocolor, but in this example, we're entering in some expression information too, and it'll become apparent why. And furthermore, they're entering in information that will also affect autocolor's reasoning about conditions in the pipetting of the elements. They're going to put methanol in, so that does indeed affect the kind of choices that would be made in autocolor. So now, at this point, they bring up autocolor, and they can choose from 10 different reagent vendors that these markers will be gathered from. And autocolor will then proceed, and it'll go in and build up a top 10 list of stain sets or staining panels. And it'll do that based on the instrument that you entered first, obviously the markers and the available reagents from all of the reagent vendors that you've chosen. And it will also it'll also consider the cell expressions. The fundamental requirement is the markers and the instrument. The cell expressions is gravy, and it's a very important gravy, and as well as is indicating the presence of methanol in the study, or in the assay. So at that point, the user can choose what, based on all the reagent vendors, is the best staining panel for those markers. And it's interesting as we work this through with bench scientists to see how this does compare with what they would have used otherwise. In the old days, before the last anniversary party, it took a while to get to this point. You had to enter in all your reagents first that you have in your fridge, and then you would basically, with the guidance of the genie, spell out the staining panel that you're most familiar working with. It would give you no optimization or give you no, well, this could be better reasoning. So that's what happens, and furthermore, from the reasoning it does to discern what the best staining panel is, it also has enough information to judge whether or not you should have controls for certain parameters or not. So in this example here, the genie automatically puts in, indicates a need for controls, and from there we have a complete staining panel. So that's autocolor, and that's just a quick guide through it. And I could actually at this point call up one of our scientists, Condola, if he's here, and we could run through, oh, there he is, sorry. And he could drive the demo through. I'll just bring up the actual genie itself on his account.
Can I ask a question?
Certainly, yeah, please intercept.
Does the program have a provision where you can put in scientific community input and like the quality of an antibody? If people like a particular staining that they get with the anti-CD4 from eBioscience is better than BioLegend, can people annotate some database that says, you know, I like this particular antibody or don't like this particular antibody?
Yeah, there's places to provide comments throughout the staining panels, and
Is
Go
that
ahead.
taken into consideration when you set up your priority of staining, or just based
It
on
will look at
input
clones,
antibody?
yeah, it will look at clones too, but it's not actually going to reason about your comments or your judgments, but it will look at clones in terms of cross-reactivity issues.
Good, if Ken tells us that that's really.
We're looking, yeah, this is more like, this is not a finished product, this is being presented for the purpose of gathering more input like that, but we've had that request before. Could you, do some basic reasoning on, I like this one better than the other? We do have the basic rule mechanism that you can put into play called conditions. And with the one example I showed you here, I chose a condition called methanol. We could have a condition called favorite antibodies, and then it would guide the genie to look at some antibodies but not others. And then that would get shipped with the staining panel when you hand it off to a student or another scientist. So, and then in that you could provide the comment why I've excluded these clones and why I haven't. So we actually do have that full capability. It was built as a side effect of building the rule definition engine for methanol and for Titon and other conditions. But you can basically define any rule you want with that, including I don't like Q dots, or I don't like, I like these eBio clones, but not those. Okay, so let me bring Condola's system over, see if I can fit that on the screen. Might be a bit hard to see it. All right, okay. So,
It's there, you just have to move it.
yeah, I know, I'm gonna put it all over. So here's the software, and when the software starts up, I'll let Condola take over in a second. I'm just going to point out that when the software starts up, there is a to-do list, and this to-do list is with respect to building an entire experiment, building your entire pipetting plan, and then shipping all of the information off to the instrument so that when you're working on the instrument, all your metadata is pre-labeled, your tubes are pre-labeled, allows you to set up plate layouts. It does numerous things, and I've only focused in my slide presentation on the autocolor. So I'll let Gondola take over from here, and I'll drive.
Okay, thanks, Stephen. So, like Stephen mentioned, this is going to be the first window you would see once you log into your account. And as you can see that the change target species, it's already been set to human because I mostly do my experiments with human and we are using my account, so it's set to human. But you could always change it to any of the other species if you are working on any of those animals ever. You could choose those and use them for your experiments.
Right.
So let's start with a new protocol. So, can you hear me now? Can you hear me now? Yeah. Okay. Beg your pardon. Okay, so start protocol, and let's start with a new protocol. And you may have to move this. Yeah. Okay. So I don't know how many in this room have seen the previous versions. If you have, you would remember that we had to go through several pages to complete all the information that's currently there in this window. So Lee likes to call it the work pages or the workstation, whatever you would like to call. And this enables you to set your whole experiment without having to leave this window ever, and not have many other windows pop up while you are defining your experiment. So if you see on the top, you have 4 tabs: the subject columns, the sample columns, the cell count columns, and the staining columns. So for example, I'm going to give an example of the kind of experiments that I do with humans. Let's say, for example, I have a few patients or a few subjects. I'm going to take their blood and make PBMC and either do a treatment to the PBMC in vitro. For example, I'm culturing them and stimulating them. I could put all that information at this stage and define my experiment, and that information will be carried over to the fax instrument when you export the experiment. So if you're using a DEVA machine, and if you have not planned out the experiment, what we typically do is once we log onto the machine, we define our specimens or tubes, and we enter all the information there. And if you're working in a facility like Stanford, which charges you by the hour, or the time that you spend there, considerable amount of time could just go in labeling your and setting up your experiment. So all that can be done remotely, not being on the instrument, and you can plan it at any time, even at your home. and then you export it when you're ready to do the experiment. Or you could plan the experiment and save it for a future date, copy the experiment, duplicate the experiment if it's a kind of a study that you are carrying over several months, like a clinical trial. So let's go to the subject column and
Just ask a quick question?
yes.
It's really naive, how you export that to
As we go through the whole process, there's a facility to export to instrument. And you notice, I mean, even in Stephen's presentation, and we can't actually see the lowermost panel of it, you can choose your instruments before you set your whole experiment. In fact, you have to do that, otherwise it won't be able to make the combination for you.
It's more naive even than that. You just send it through the network?
Yes,
Walk up with it on a memory
you could do
stick?
both.
Oh, okay.
But at Stanford, we do it through network.
Through many firewalls.
Okay, so subject identifier, I'm going to select that, and you can just type in your subjects. Let's say we choose 3 subjects, subject 1, 2, and 3. So you could just type it in there, or they may be available because I've demoed it many times. That's fine, 2. Okay, 2 are fine.
Right. Subject.
2. Okay, so let's say we are starting an experiment with 2 subjects. And like I said, if you see on the bottom, there is, it's just 2 different views. Maybe you want to show how to, yeah.
All right.
You could either see all these different tags. There's a facility called tag, which is essentially a qualifier for either the subject or the sample. Tags for a subject would be like whether the person is a patient or age or sex, things that you would not expect to change between the experiments. But sample tags could be like once you take a sample, whether you're treating the sample, whether you're culturing it, or whether you're stimulating or not, anything that you would do with the sample. So you could define the conditions of your experiment, and then the software can apply combinatorial process and apply it to all your subjects. And I'm going to demo that, what I mean by it. Let's say I'm starting with these 2 subjects, and I isolate PBMC. And I'm going to culture PBMC at 2 different oxygen levels, and I'm going to have 1 control, unstimulated, and 1 well with stimulation. So instead of having to create all those 4 conditions for each of these subjects, I could define my conditions that I'm going to apply to my samples and combine it with these 2 subjects, and it will automatically create all the samples for you. Okay? So in this experiment, my sample tags would be, for example, stimulation. So I'm going to check that in. You see stimulation because I've already entered them. So if you are starting with a fresh account, obviously these things are not going to be there, and you can put in whatever sample tags you would like. And let's say, and pick anything, like oxygen levels, for example, on the top.
All right.
Okay. So now you could enter the values for the stimulation and oxygen right here, or we could even go to the previous view we had where you saw a small table at the bottom, and those are just 2 different views for you to work with, whichever you feel comfortable with. Like personally, I work in this view. I add all my subjects and the sample tags at the bottom, and then I can apply that combinatorial feature to apply it to my sample subjects. Okay, so 3. So if you are taking cells from these 2 subjects and you are either stimulating or not stimulating them at 3 different oxygen levels, all you need to do is to select everything and then click on the button on the right there, and it will combine these conditions to those 2 subjects. So it's going to ask you, do you want to apply all combinations to produce 12 samples, or add each tag once to just produce 6 samples? So whichever way you're doing the experiment. I'm going to take the cells from each of these subjects and apply all those values, because that's how my experiment is set up. So you say yes, and it makes the samples for you. And then you can go ahead and go to the cell counts tab, and like Stephen mentioned that initially when the software was made, it was made like a planner, experiment planner. You can choose what your initial count was, what your final count is going to be, how much sample volume you want to add. All that information can be input here. And these subject tags and all this information can then be exported to the machine. So you can either arrange your samples to be your specimen in a DIVA software and each of the conditions to be the tubes, or you could take the conditions to be the specimen and the subjects to be the tubes. You have the facility to change everything in the final layout of the experiment. Does that make sense? Yeah, okay. So this is about the planner. If anybody wants to play around with it, definitely you can get an account. I'm not going to spend too much time on this. But let's say this is my experiment and I'm done with it. I now have to select my staining panel to stain these samples, right? Now I have two choices. If I already have staining panels that I have used for other experiments, I could just import those to this experiment, or I could start fresh. So I'm just going to show add prior stain set if you click on that. Now, these are all the different stain sets that I have made. And some of these stain sets are made for LSR type machines, LSR2 type machines, and others could have been made for, let's say, a Cantor or something. If I have LSR2 selected as my default instrument and I'm trying to apply a Cantor combination, as long as it can handle it, it would take in that combination. But if I made a combination for LSR2, which is an 3-laser or 12-color experiment, and I'm trying to apply it to an instrument that has only 2 lasers and can handle only 6 colors, it would warn you. It will not do it, because there's no way you can do the experiment. But if you are, let's say you choose an experiment for LSR2 and you have several LSR2s in your facility and the particular instrument for which you have made this combination fails or something, it will let you export it to other instruments that have the right filter set and the configuration. Otherwise, it will just not export the experiment. So that's a great check before you actually go and do the experiment. Okay, so I decide not to take any of these combinations, and let's build a new stain set just for the purposes of demo. So we are back to the same window that Stephen had shown before. There are 2 views again. These are all the antibodies that are currently there in my reagent book, because I have already done experiments and I have imported these reagents into my reagent book. So I can either see them, or if you are starting fresh, you are not going to see any of these reagents because there's basically nothing there. So then you would be seeing this view. And now you can build your staining panel. So let's build a simple staining panel. That's a CD3, CD4, CD8. And then let's say we put 2 antibodies, CD19 and 14, as our dump. I know the B-cell people will be offended, but that's fine.
CD19
Yeah.
and what was the other one for the dump?
CD14. Okay, so you notice that when CD19, when Stephen created, it defined it as dump. The dump channel actually populates and it says dump. Okay, and if you see the other columns, immediately to the right of the marker is a column called Wiki. If you click on any of the buttons, it'll take you straight to the web and show you everything about that marker that you would want to know, any alternative names or what the specificity is and things like that. To the right of that one is the target cell expression. The target cell expression, you can choose one particular cell type that you are focusing on, or let's say if you're working on whole blood, you could have several target cells that you may be intending to study in that combination, and you can define your expression on each of those target cells. So obviously I'm interested in CD3, CD4, CD8 basically. Those are the 3 markers because I've already dumped the CD19 and CD14. So let's say I define my target cell expression to be for CD4 T cells. So I click on the target cell and I say, I can label it as CD4 T cells. That's okay. Yeah. So now, if you see, it automatically populated the markers there based on how I had earlier defined it in my previous experiments. But you could go and change this expression. Let's say there was a problem in the way I had defined that CD3 is actually not very high on CD4 T cells. So you could click to go at each level and change the expression. Again, this expression is more based on visual appearance when we use a particular antibody, whether it stains brightly or not. It's not really counting the actual number of molecules on the cell. Okay. So once I define my target cell expression, when it chooses the combination, it takes that information into consideration in making the decision of what kind of markers it would want to choose. Like for a for a target that is expressed very low, you may want to put a bright marker rather than choosing a dull fluorochrome on a low expressing marker. And let's add another condition to this experiment, that I want to look at a phosphoprotein, like say P38 or something in these T cells. So now I can add another marker here called P38. Can you enter a new marker? Okay. So here's my experiment. I'm working with PBMC and I'm stimulating them with, let's say, a reagent X, and I want to see whether P38 is getting activated in CD4 cells and CD8 T cells. Okay. So I chose my panel, CD3, CD4, CD8. I'm gating out my B cells and monocytes, and I'm also going to look at P38 expression in all these cells. But I can now tell the program that P38 is going to be after permeabilization step. And it's critical for phosphostaining because invariably we use methanol, and most of the protein-based fluorochromes don't survive the methanol step. So this instrument will not choose any fluorochromes that's going to compromise because of your methanol step. So when you do autocolor, the way to do that is you check your methanol column and it will populate a column to the right which tells you what kind of a condition you are applying to your experiments. And it's already showing that all the protein markers like CD3 to CD14 is before methanol, but P38 is after methanol. Again, because it remembers this information from my previous experiments. But had this not been there, you would have to go and tell that, okay, these steps are going to be before my methanol step, and P38 is after my methanol step. It doesn't have to be methanol, it could be any permeabilization reagent. And let's say you take tridon or saponin, then it's not a problem because saponin doesn't really compromise your PE and APC-based stains if you happen to stain it before permeabilization. But methanol would kill the protein-based stains. So then it would know that none of these surface markers should be put on either PE or APC-based fluorochromes, but it should choose other fluorochromes that could survive the methanol step. Okay? So let's say this is my staining panel. Now you go to autocolor and hit optimize. Okay. And I'm interested in seeing my top 10 combinations because I'm going to choose a combination from there and hit proceed. Yeah, we're pretty much done. Yeah. So, okay. Take this. Yeah, you can just. So once it calculates the top 10 combinations, it shows there in the button that you have already calculated, you have your top 10 combinations ready. If you want, you could go further, but and then you could pick any of those top 10 combinations if you have a particular preference.
You don't have your top 10 yet because you stopped it at 2,000.
Yeah.
You've got top 10.
Amongst those calculated, exactly.
Well, no, the button lights up when it's already got perfect scores.
If it doesn't light up.
Oh, it doesn't.
Yeah,
you have to let it go. If it doesn't light up, you have to wait until it's.
So, I mean, because we are speeding up the process, I'll just stop here, but while you were making your staining combination, you have those columns for clone and fluorochrome. If you have a particular clone that you absolutely want to use in your experiments, you could lock that clone. So Ken, to answer your question, currently you could either choose antibodies to come only from a single company, like BD only or eBioscience, or if there's a particular clone that you are interested in, just lock that clone. So it will build the stain set around that clone. Or you could do that using your fluorochrome, and it'll build a stain set. And if it can't, it'll tell you that you cannot make a combination with that clone and color locked up like that because the reagent is not available. Okay. So.
All right. Well, that's autocolor in a nutshell. All right. All right. Does anyone have any questions?
Can you also fix what reagents it uses by putting them in your notebook or whatever you have there on your list?
You could do that, or you could just lock them right here. We haven't shown you all the screens, but you can say, I know exactly what I want for the 5, and the 2 I don't know about. Have fun and come up with the best combination. So you can lock down, or you can even set a preference lock, saying, I prefer this, but, let's see what else you have for me.
Yeah, it also, you had mentioned that it knows about what you have. So if you actually have purchased reagents and they're in your fridge, it knows about that and it'll track your inventory on
Right.
those to know whether you're running out or not. But you can tell it to use only the ones you have in your fridge,
Yeah,
and then that's what it will do.
yes, that's another good point. So you can narrow the search either by just locking an explicit conjugate, irrespective of whether it's in your fridge, or as Lee says, you can lock it to everything in your fridge. Or you could come up with a fancy rule about how to lock things. No Q dots, no. Go ahead.
And when it's searching all the different databases and reagents that you've loaded, can you also ask it to find the cheapest one or the?
Not at this point. We've had that request often. Those are easier things to do than what we've been doing since the last. The question was, can you tell a genie prefer the cheaper reagents? And yeah, that's not a hard thing for us to do compared to some of the things we've done.
It's hard to get the manufacturer
To give us the price,
specify what
yeah.
price they're charging you.
Different depending on where you are.
Yeah, and essentially what we're calling our top 10 display is just based on spectral fluorescence and cross-reactivity of antibodies, and that's how we rank it. But the display mechanism allows you to reorder and resort things according to a number of columns, and so one of the columns could easily be the total cost of the stain set, or the total number of reagents that are not in your stain set versus, you could sort it any way you want, and then have a secondary sort on the fluorescence ranking that we gave, or the cross-reactivity ranking. And then it would remember that you wouldn't have to reteach the window every time you want to look at the top 10 stain set. So this particular portion of the software has certainly taken a lot of our efforts, but at the end of the day, it's probably only going to be used frequently by those who are working for the facility and advising scientists on their staining panels. The average scientist may not, be creating a new staining panel every week unless, it's Steve DeRosa or someone like that. But, so there's a lot of work here, and we foresee that it's going to be the people working for David and Marty that are trying to help scientists set up their fax run that will find this most valuable. Everybody will find it valuable, but
Maybe we can eliminate the need for those people, that would be great.
need for that portion of the job.
Yes.
All right, David, I'll get your slides on.
Well, let's see what else we're having. Let's see, Gary Nolan was wanting a slot here. Do we have,
He has to arrive.
well, he does, he's got avatars, doesn't he?
Probably.
Do we have
Try your
any?
email.
Okay.
We'd like to hear about the auto compensation.
Yeah, okay, well, something else. Okay, right, well, if there's anyone else have topics they'd like to bring up, and
I was just wondering, Dave, you said there's so many people coming with all different kinds of assays using the machines. How do you put people connected with, let's say you want to do something and hey, someone else was doing this assay and maybe you could talk to them. How do you link people based on the kinds of applications they want to do to kind of help each other?
at this point, it's just the experience and memory of the facility staff. We don't have people give us some information about what they're planning to do, basically on the biosafety kind of aspect that we have to screen and see if there's any concerns about the work. But that kind of stuff doesn't get entered into some sort of database or anything about being able to match people up and such. And also a lot of people who, what they sign up as their initial thing may not be what they're doing a year later either. So yeah, that's helpful, and we do connect people that way to the extent we remember and know who's been doing what, of which the facility support staff, who partly do some actually operating for people and partly just consultant, and also just back people up if they're having any trouble, and there's a lot of that going on, so there's a lot of that understood, but it's not as organized, yeah.
Can I add to that, Dave?
Sure.
Yeah, Vernon actually started me on all of my interest in using the internet to support experiments and things, like something called Science Exchange, which still
Okay,
exists as a website.
So do you want to bring up the
in our
one with
mind,
the,
and still
okay,
exists
that's the
as something
compensation
that can be
part.
done.
David, go up, Mike. David, go
And-
up, Mike.
Do I need to go on?
If you're, yeah, if you want to get recorded, you have to.
I don't know that I want to get recorded. But that's, the idea of science exchange is something that now is becoming possible with the current forum programs. And so once I get through with this, one of the things I want to do is put up on the Cytogeny website, which is actually the Woodside Logic website, a forum, and you can search forums for things, and people can set up, allow people to set up whatever kind of forums they want to set up under that situation. So you can set up a forum for people working on chlamydia, or set up a forum for people who are working on some kind of new fluorochrome or whatever, but you can draw people together. And I think that what actually is going to happen is in the next several years, things like Facebook will become, what we need is a Facebook for scientists to use to find these kinds of things, and have our so-called social interactions be our work interactions. That's going to happen. Somebody's going to offer us one of those. I don't think we'll have to build it. And I think that within the next 2-3 years, that will tie in, but we will tie Cytogeny into that as quickly as we can, so make it so you can pass the protocols around very easily for stain sets that will work for particular diseases or for particular kinds of cell types. Davis.
Cytometry has a publication type devoted for publishing multicolored panels.
It's not publishing the panels that's any good. It's actually, as far as I'm concerned, because you really have to know the people behind the panels and
But
talk
it's,
with them.
so it's specifically designed.
Yeah.
So we created this specifically for this purpose.
Okay.
So the publication type specifies that you have to show that you've optimized the panel. You have to show how you optimize it. You have to show the complete range of all reagents that you've tested, why you chose the particular reagents, the particular color combinations, and then show examples of that.
Yeah, I really want to link into that. I mean, I know.
So that was created about a year and a half ago, and there have been about 4 published so far, and we're soliciting many more. But they're called OMIPs, Optimized Multicolored Immunofluorescence Panels.
Yeah, my problem with that is just all the things that you have to publish in it, which some people have and some people don't. And what really gets asked for, to me more often than not, is something that's pre-OMIPs that has to do with, is somebody doing this I can talk to, who,
Right.
has been working on chlamydia. And it really isn't exactly the stain set that we're chasing with. And the problem with the optimized stain sets is that it's okay as long as you really have an instrument that does that. And most people don't know whether their instrument can actually do what the OMIPs can do. So what we need to do is essentially put the OMIPs into here and determine the...
And that would be great. You should do that.
So we should do that.
The idea is somebody should build a database into it eventually.
Start the other one.
Because the goal should be, I want to do a staining with the following 5 reagents. What have people done with those 5 reagents? And there may be more complicated or less complicated
Yeah,
panels.
and we'll have all of that to the extent that people are participating in using the Cytogeny stuff.
Right,
But
but
we
the
certainly can do the
Yeah.
OMIPs thing of putting the OMIPs stain sets into Cytogeny and then allowing people to choose them if that's what they want to choose as part of the things they do. And they can use that as starting and put their own reagents in it and keep on going from there.
So there were 2 reasons behind doing this in cytometry. One was to provide a peer-reviewed, or suitably somewhat peer-reviewed resource for this material. The other was to provide a mechanism by which the people who have spent all the time creating these panels can get some recognition. So it really came about because the postdocs in my lab were saying, we keep getting asked for these panels and we keep giving them away, but then there's no mechanism by which we get cited for them. So the idea was that if we publish these, and these are real publications in cytometry, people will now cite them and reference them, and then there's a trail that now provides a mechanism for recognition of the hard work that goes into these.
This is a good workshop because I hadn't thought about getting the OMIPs connected in, why it would be really valuable, and I could see. So let's just take the other on it. I I mean, I think that there's no reason why it can't be done. And as soon as we get our resumed act together, we'll do it.
Okay.
It's actually-
All right, then I'll pick up with a couple of things here. You've been welcomed by our state rodent, the California ground squirrel, a prominent campus resident. And something that's taken up a fair amount of my time in the last few years is it's been my wife's quest to visit all of the U.S. national parks, which she completed 2 weeks ago, all 58 of them, including 8 in Alaska, American Samoa. So it drags you around a bit to try to do that. So anyway, that was good. But so we just got back from visiting the marmots of Rocky Mountain National Park as they're hurrying to store away some food for, and you can see getting quite fat, which they'll need because of the very long winter up here. Coyote came to hunt mice right near where we were having dinner at a picnic area. It was around lots of beautiful tiger beetles. This, we think, is not radiation-induced. This was at the Rocky Mountain Arsenal, north of Denver, which is now a wildlife refuge, but is a very toxic site. But it's the Rocky Flats is the one where they lost all the plutonium, which is not some distance from there.
They also have bad cantaloupes there.
Yeah. But anyway, there, and this is the famous great sand dune tiger beetle. So you can tell he stilts himself up from the hot sand. And it was getting to be fall, so all the males were getting all revved up for the fall mating season, making fools of themselves largely because the females mostly weren't at that point, and the elk were trying to herd the females around and things like that, and they were not having much of it yet. But that was good. But the tarantulas were also out migrating. The males migrate to find females at their burrows and stuff in the fall, so they were out, and very nice. Went out and saw the goblins. In fact, you can see just a little hint there that the ravens treat the goblins the same way the pigeons treat statues. So that was, and with the Goblin Valley State Park, other thing, of course, in these travels, we go to 2 or 3 national parks in the course of one of these trips, but we end up going to a total of 20 different places and spending, 4 weeks doing it. So that was, got around. And some really strange, interesting formations up there. Same place as the Goblin Valley State Park, Utah. But anyway, while you're, and yeah, that was not a whole lot of fall color this last month in the Rockies. It's largely Colorado, and stuff, and not a lot. But Rocky Mountain, Great Basin National Park in Nevada. The aspens were going crazy with apricot colors rather than just the typical gold of the main Colorado Rockies, so it was neat. And if you've got a little time on campus, right over in the cactus garden, over near the mausoleum, there is a ceiba tree in bloom. It was a week ago. Beautiful thing with an amazingly ornamented trunk. This is the sort of side view of the trunk of that tree. So anyway, that's worth getting to. And of course, if you get farther, have a little time to get farther afield, over on the coast, we got anemones, sea urchins, lined chitons, stuff. So anyway, and of course the harbor seals. So anyway, enjoy. This is a great area. Okay, so I'll go on to just a quick rundown of a couple of topics then. One is we have gotten into recently in the MAX facility, recently got into small particle detection. Jennifer Jones in particular is working with microparticles and exosomes and such. So she was wanting to be able to work with these and sort them, and actually one of them, and had the systems going to do the rest of it. And so we checked out several of the facility instruments for this purpose. First thing, here's data. The influx turned out to be the easiest to work with because the sheath and sample stuff is completely separate, and we could put in a much smaller filter to filter the sheath fluid and avoid getting problems in the size range of the things we're interested in finding, and be able to handle that easily and just change the sheath pressure as required, because these fine filters hold up the flow somewhat, and you have to change the pressures. But you can see it really worked, that the filter, the standard 220 nanometer filters, 0.22 micron, and gave the upper kind of displays in scatter from conditions, and in once the 100 nanometer filter was in, that went way down. So that was helpful. And let's see, is there actually a pointer here? We had one earlier. Just testing this, testing with different size plastic particles, you can see that in forward scatter, this system, the influx also, the other thing is it was intended partly as a plankton system. So it has special forward scatter optics, high collection, yes, Dr. Nolan has arrived. Okay, we'll put him on as soon as we can get this going. Yeah, so anyway, you can see that we could detect with this system that had stronger wider collection optics, a fine aperture, and a PMT rather than the diode detectors that's often in forward scatter. The noise level was such that we could easily find 300 nanometer particles of that, but 190s were just completely lost in it. And whereas by side scatter, the noise level, electronic and background light noise level without any particulates was in this range. So even the 300, even the 190s are way above that, and potentially we can find them pretty well. So in terms of those results, then looking here, and by triggering in green, in a fluorescent particles, we can see that the 100 nanometers even stand out in side scatter somewhat above just above the noise levels, where this is just sort of random triggers, basically. And that the side scatter can't really be used to trigger on it because there are enough frequency of signals up in that range so that 100 doesn't work, but 200s are way above background. So anyway, it looks promising to find things in that size range. And I'll just mention that well, of this kind of particle, and I'll come back to the biological particles right in a bit. So we find forward scatter drops like fifth power, over fifth power of, so it drops out rapidly when things get small. And the side scatter doesn't drop nearly as fast. So the 200, 300 nanometer size for forward scatter, to be able to really find it by forward scatter, you need 300 or so. But the side scatter, once the extra particulates and stuff are low, you may find quite a bit smaller. And this is checking on LSR2. The flow system there requires some more modification to be able to put in these filters that drop a couple of PSI across the filter to get the flow you want. Or possibly, they haven't even tried just putting in highly filtered sheath and see how much that does. But the red here is background particulates coming through the 220 nanometer standard filter. And you can see, because the 190s are uniform, we can find them very easily as a big spike in that distribution if we put them in and mix. But you couldn't run them at a low frequency and find them well. But certainly in side scatter, they're way above background. This is using what would normally be the PE channel as a 532 nanometer side scatter. So part of what I was doing was checking the correlation between the 488 side scatter, the standard one, and this, because different lasers and different situations might give you different sensitivities, and they do. The LSR2, because of its standard type detection and has a much noisier forward scatter channel, so these 0.8 micron particles are barely above threshold, above the noise level in forward scatter. So we would have to do something more with forward scatter on that instrument to have it be useful for really small particles. And there's one of the things we might get into is redoing that optics to put good detection in one or more of the LSR2s, and but that depending on the interest and need for it. And the LSR2 gives, for side scatter, gives the similar 2.5 power particle diameter versus side scatter signals, and indicating that finding them down in the 100 nanometer range is probably quite reasonable in side scatter. Reflecting on the different lasers, in this system, you can see that in any of the 190 nanometer particles are up here. They're way separate from the background in the 48 or the 532 signal. A lot of what's going on coming in as real particulates is correlated between the two here, and there's something else going on in the blue, because we got this that doesn't show up in the green, which is a fairly separate laser. So if there's a correlation, it's very clear there's a real object there. But for instance, the red laser, the noise level in this dimension is the red laser. The noise level is not a whole lot below the 190 particles, and in the violet laser, same thing. Converting a channel to be a side scatter channel in those lasers isn't very effective. So evidently the noise levels of those lasers and such is getting into it, and we can't do it. But if anything, the 532 is better than the 488, and there's a big gap there between the level of the 190s and the real noise level in the, so potentially, again, we could do pretty sensitive work, especially with the 532, it looks like. And the checking an ARIA 2, again, the 200 nanometer particles are within the background objects that we're seeing, but they're above the regular noise. So cleaning up that background, certainly those at least should show up. The 100 nanometer particles in side scatter are just way down in the noise, though. So Jennifer's project, one of the things that she has brought in is that she has access to a nanosite device. This is for sizing very small particles. And so what it does is it's a microscope system generated on camera, generating images, and it runs a very thin ribbon of laser light in the focal plane of the camera, and just sees particles, and light scatter off those particles is detected by the camera and just analyzed as video for, okay, quick time, no. Okay, it's not going to come in. Moving movies from one computer to another is always risky. But anyway, so we lifted a little thing from the website of the company there. They had larger particles and smaller particles mixed in showing it. And you see a field where what happens is the big particles kind of wander around slowly. Those very small particles jitter around a lot. And it's the analysis of that video that allows you, for regular Brownian motion, to calculate an estimated hydrodynamic size of the particles. And so it does that on a particle-by-particle basis. So it just samples particles, tracks them for a while, gets an estimate of their size, and adds them into a histogram, so that in this case, on the influx, we have the very low forward scatter, noise level really on forward scatter, and then the ones that are showing up a little bit above the background in forward scatter on that tuned sensitivity channel. And we find that the ones from down here, the sizer shows a histogram in the red, and the ones up here, it shows the histogram in the blue. And putting those sorted fractions onto an electron microscope, we see in here that we have the the ones that on the high side here come out as microparticles, and the ones on the low come out as the exosomes. So it seems that we can pretty reliably separate those and begin to work with them, the systems. Okay, Stephen, you want to swap over to that other one? Combine things from a couple of different PowerPoints that actually we can, yeah. So I'm just going to go very quickly through the.
Quickly.
Yep.
We gotta get
Okay.
there.
Okay. So in the there's a poster up tomorrow. Kate Boheim has assembled one, although I think I'll have to take care of it. She's going to post it tomorrow on the automated fluorescence compensation, which there is a program that people can actually set up and use to do that, and that's what the poster is about. The background on that, very quickly, come back here. Okay, slideshow. Okay, oh, it is up. Okay. All right. Okay, so we have the objective here is to do automated analysis and provide information, quality control information, things we've never had before in compensation evaluations of, is this really good? Is there something wrong with this, any problems and things with this sample? Confidence intervals on the matrix values that come out, as well as some other information, estimating flow electron scales, which are kind of interesting to know and potentially useful, and understanding more about background and noise in the samples. And the photoelectron scale facilitates the potential for doing synthetic FMO evaluations, that is, finding, estimating from fully stained samples the FMO, expected FMO distributions. So let's see, how do I persuade this to advance? The right arrow is not talking to it.
Down arrow.
Down arrow.
It's a down arrow. Oh, okay, that's,
Oh, did it go?
let's see, well, it changed on the screen here, but it didn't change up there.
It's fine work.
Yeah, okay, let me just, yeah, okay.
Let me see if I can get a mouse over there and just underneath that. Oh.
Yeah, and that was, okay, so Wayne Moore and I developed a model for the kind of things you should expect, and that allows us to look at the data coming in and evaluate and see if there are events that fit the expectations for what a single stain compensation control should look like, and if not, clean up the data points that don't look like they're part of that, and then do a fit and an estimation of how good that fit is. Just things like the fact that we expect that the brighter a single stain is, the narrower in a log-log display it's going to be, and that pattern, and building that all into a model rather than simply gating a region and using means or medians or something of the events in that region as a calculation of actually weighting the different events depending on how bright they are and how tight how uniform we expect them to be, how well they should be fixed in space. So that potentially doesn't necessarily give you different values, but it's the kind of thing that allows us with the model to actually estimate quality factors and uncertainties in the measurements. And so the variance has a linear relationship with signal on the primary channel detection, for instance, is a large part of the key thing in the model. And then the data gets groomed, scatter-gated, getting out the events that in all the colors, it examines in all colors so that events that are out of color in one or two channels or in channels that you normally wouldn't look at can be used. So in this case, since Wayne developed using nearest neighbor techniques to do scatter gating, estimate a central population of scatter on a very general basis so that you can usually get a good starting point and get rid of a lot of the really outlying events that way, and get a distribution like this, and then working mostly from an unstained scatter distribution, then go into the rest of the system from there. And then in terms of pruning the fluorescence side, it, for instance, detects things like the red events here that most people would just put a box around and say fine. In fact, when you look at some other channels, like way over here, you see those are anomalously high. There's something else going on here. And because it's working in the, looking for problems in any dimension, something that doesn't match the main color pattern of these in all dimensions gets screened out. So that helps it throw out really bad data and also subtly bad data, things that are, right? And then fits a line to that from which the matrix elements are derived. And it's actually very sensitive. In this case, we've got an unstained and a stained sample, a lot of just high background cells out here in the same brightness range as the real stain, which is more or less these over here that are stained but not down the diagonal. And the model latches onto those very nicely, and it sees that these have a very different overall pattern, and that these have lower overlaps, essentially, in other channels. But anyway, that these are really different, even though they're there, and it doesn't get confused, even though there are more of them. So anyway, I'll say one more thing, just to people who have things like LSR2s, something I've worked up and noted a while, that I've been analyzing the instrument behavior and trying to optimize the situation so that we can set up standard starting conditions that most people can run at. The data looks uniform and everything, and you don't change that unless you really need to. Unless you're running things off scale, you know that you're, in general, set it up so that you know that is at the low end, anything that you can distinguish on this system from background, you will be distinguishing. And then only if you have stuff running off the high side do you have to go to a non-standard settings. But in the process of doing that and evaluating the instruments, one of the things it found was that if the PMT voltage is off, the electronic noise has a certain value, and the DIVA system is often between the 7 and 15 range, whatever. And ideally, when you turn the voltage on a PMT but don't give it any light, the noise should stay the same. A PMT should be totally quiet without noise, and we can then estimate the rest of the behavior of, in this case, very unfluorescent 1 micron particles and unstained lymphocytes, and see how they behave and choose running conditions from that kind of data. But I was finding in a lot of channels when I turned even moderate voltage on, the noise would go up and would continue to rise slowly, not like gain of PMTs, which would rise fast, but would still continue to go up somewhat and would compromise our ability to detect low-level signals. And in fact, the newly delivered LSR2 with the UV laser that we got recently, 2 of the channels were giving standard deviations in just voltage on noise, light blocked, over 100 units, which is, really messes things up. And a number of them were kind of intermediate. And it turned out that basically the contact, there's a design issue there partly, and the contacts, if you take them out between the PMTs and the preamps and that, and just take all the contacts apart, put contact cleaner on them, put them back together, the noise is flat, like that first detector. And your ability to detect low-level stuff is helped by quite a bit. So just a warning on that kind of thing. And the other thing I've noted and just started pushing on a little bit is that the DIVA-type systems, they give you a width signal that's really just the ratio of the area to the height signals on a channel. And those are kind of squashed up and they don't really look like much, and people will often just put a height versus area plot up and do diagonal gating off of that to do double exclusion, things like that. In fact, doing a computed dimension that expands that out and really looks at it, you can see things like live dead that overlap some, the width doesn't overlap. And so that's really worth looking at. And it distinguishes things like 3 micron particles from 3.6 micron particles, even though the laser beam is 25 microns wide. So anyway, why don't we have Gary talk? And while he has a minute, if there are questions or
Dave,
discussion points of that sort. Yeah.
So about your last comment about looking at that noise, so in order for us to look at that on our instruments, do we just turn the lasers off or just block the laser light?
Yeah, for the channels, it's not your threshold channel. You can just put a paper card in the laser.
Or something in
The thing I've done that actually works really nicely is you take a reflector, a blank, empty reflector for the octagon or trigon system, and put flat black stuff in it.
Oh,
A null reflector, and that blocks the channel behind it and doesn't reflect to the oncoming ones. And so you can leave everything on, just pull the first reflector out of an octagon or trigon, drop in a null black thing, and then see if things change between 0 voltage and your typical operating voltage. And if your noise standard deviation of that goes up, that's not good.
You just clean the terminal, clean the actual connector directly from
Yeah.
PMT to that connection.
Yeah. So use contact cleaner on, clean them up, stick them back together again. Or actually, usually if you just take it apart, wiggle it, and stick it back together, it'll help.
And what was that computed parameter you were just talking about?
Well, it's the width, and it shows up kind of squashed up, not using much of the nominal range, just the way they've chosen to display it. But if you just take, like in FlowJo, if you take a computed dimension that subtracts the space that's before anything is happening, and then scales the next 30,000 or whatever out of the 200,000 into a full view, the resolution is really there to see more than you can see in normal. And we have Dr. Gary Nolan set up. All right, you're on.
Thank you. Okay. The scary thing at looking at what is still being done to make sure that we get quantitative information that's well QC'd off of a flow cytometer 30 years after I even began, which was 10 years after, and it's still going, makes me really wonder about what I'm going to show you now. So CyTOF, I'm sure you guys have all heard a little bit about it. And so I'm just going to dive right into it without all the usual preliminaries. So we, at least in the lab, have been working with, how does this thing work? There we go. Have been working with primary cells and signaling at the signal cell level. And the objective has always been to work in situations where you would interrogate a cell, right? The interrogation process itself is about stimulating signaling, and that we would always compare what is normal signaling versus what is pathologic signaling, and that often you can't actually even reveal what pathologic signaling is unless you ask questions. One of the jokes I often use is that somebody comes into the room to interview with you, and so you sit there and do you just look at them for 30 minutes, right? Or to sort of measure their basal state, or do you ask questions, right? And you ask questions, of course, because that reveals something about the makeup of their mind, right? And that you're going to be drawing inferences about what they answer. Now, the other interesting thing about asking questions of cells is that I could ask the person who comes into the room, how are you today? And if they break down crying, right, I know that it was neither the question nor the answer that reveals the fact that there's a pathology, right? And so the fact that there's a pathology is by virtue of the fact that the human brain and other systems exist in networks. And if you believe in network interactions, you don't necessarily have to know exactly what is wrong with the system. You just have to know that it perturbs things nearby, and that by perturbing things nearby, you can then, if you can pick up flags, for instance, of nearby elements and changes in how those flags act, you can then reveal that there is probably something not so distal from the event that you're reading that in fact is the pathology that you're interested in studying. So we perturb ourselves mostly with cytokines, plus minus drugs, primary cells right out of the blood of patients or frozen viably. And then we would fix and perm cells, stain them, go to the fluorescence activated cell sorter, and we would measure cells by cell type or by cell signal. And of course, cell signal is usually almost always in the unstimulated state as compared to the stimulated state. So we all know the virtues of fluorescence, but we all know as well that there are limits to the number of parameters that we can measure, which caused me to start searching about 8 years or so ago for new ways to do this. And then luckily, Scott Tanner from the University of Toronto found me, not the reverse, and said, gee, I've got this machine that can measure elements on a single cell basis. And I think that this could be useful for measuring single cell elements, single cell proteins. I said, well, how do you do it? Well, I just attach the elements to antibodies and replace the fluorophores. And I said, well, what do you do with it then? Well, he said, then I pass it through a 13,000 degree flame. And then I suck it into a time-of-flight mass spectrometer, which I knew nothing about. I mean, mass spectrometry I knew not a thing about when Scott first walked up to me. In fact, I was trying to get away from this guy when he came up to me because he just seemed to be talking nonsense. But when I realized that after about 5 minutes of chatting that he actually had something, that's when we send people up to his lab to try to see if this device actually could live up to the promise of what he was aiming towards. So this is the color of the machine. I'm not happy with the color, obviously. No, I'm not in the color, so I accuse Scott of being colorblind a lot. But this is actually the business end of the machine. And it's not unlike a flow cytometer in many of the functional aspects of what you're doing. It just replaces the fluidics with not actual water, but with plasma fluidics, fourth state of matter fluidics. And actually, you can just barely see the light in there. And then after that, when you collect the data comes out looking more or less just like what we think of, but now you have the additional problem of having 30 or 40 dimensions of data to study at a time. So this is the signal resolution difference that you can achieve with the machine. This is, of course, a best of class set of fluorophores, so-called quantum dots. And these are the kinds of signals that we can get out from this device. So we know this is sort of Mario's top of the line number. I'm sure people have...
You can do 18.
You can do 18 now? Great, great. The great advantage of this, of course, is that we have about 100 non-biological channels available, and with a couple of statistical tricks that we've come up with, we think we're going to be able to do 300 to 400 parameters per cell in the not too distant future. Dynamic range is around 10 to the 9th, and of course there's zero background. There's no autofluorescence because there's no automass, right? If you've got automass, you've got a problem. Maybe that's what dark energy is. So what do we use? We use elements, but we use the isotopes of elements. So the isotopes are purified. They're actually most of the lanthanides, the ones that we use in the initial series, are, and some of the actinides, are mostly actually mined in China. They're then sent to Russia, where they are then superheated into a gas, and then they're centrifuged to separate out the weights by centrifugation. They're then turned into little metal ingots of about half a gram or so, and then those are about $5,000 to $10,000 depending upon the isotope or the relative rarity of the isotope. So for instance, as you can see here, With palladium, there are several, but there's one that's relatively infrequent. So if you really had to get that, you'd have to spin a lot of palladium to be able to get at some of those lower represented of the stable isotopes. A lot of times people hear isotope and you think radioactive. These are all stable, right? No radioactivity is used here. So all of these are potential players in the system. We've actually even been using iodine. It's not on here in this range. We've been using iodine as a replacement for bromodioxyuridine. So we use iododeoxyuridine, and then we can actually measure DNA incorporation with that.
One thing to point out, once you get past about 40 or 50, compensation becomes a big issue.
Yeah, that's actually what I'm going to start to point out here, is that there are problems with some of these lanthanides in that you get a lot of noise in here. Primarily in this case, it's because of oxides that form. You don't get complete atomization and ionization of these, and then they begin to bleed into each other's weights. And one of the things I think Mario was referring to was that even though we have isotopes, they're impure, and so they have sort of a spectrum of nearby neighbors that are of a different weight and will begin to creep into the weight classes of the other isotopes that you're potentially using. Is that what you're referring to?
Basically, you can't get pure forms of some of them.
Yeah.
So some of them they're like 85%, 15%, so that's automatically 15% compensation.
Right, somewhere else,
It
yeah.
would cost more than the U.S. Treasury
To get them,
100%.
right. So that's why we think that we're going to be able to reasonably, just on a per isotope basis, get between 50 or 60. And then beyond that, it's incrementally far more difficult, which is why we've come up with these other tricks to allow us to multiply some of these by making various kinds of statistical approaches at it. So that's about a year or so away till we get that working. So there are high sensitivity channels available in this weight range, and then there are those that are less useful, but they're still measurable. You just need a lot more of them. So kind of like the fluorophores. So this is how we get the isotopes on. So we get those little metal ingots. And by the way, one of those metal ingots will last us till the end of time, at least my lab, right? Because you don't need very much to do the detection. And part of the virtue of the system is that once you've made salts of these, you drop them more or less in boiling acid, and you make a salt of them. The chelator that we're using, that was originally developed by Scott and Winnick up at the University of Toronto, has a very high affinity, right, or avidity for this. It's a 10 to the minus 16, so you don't need very much. And actually, this is also a virtue because many of the lanthanides are poisons, but the amount that we're actually using and then expelling into the environment is extremely low. It's so minute as to not be worth worrying about from a safety point of view. So we chelate, and then we, again, work with Scott and Steve Winnick to create a number and test a number of carbon backbones onto which are decorated many of these chelators and then onto which are bound the various isotopes. And actually, we tested with Scott a variety of these. It turned out that just the linear version of these was the best, all various branched and dendrimer structures that ended up actually being more problematic. They ended up being sticky and would bind to cells more than they would bind to the object of interest. So the chemistry that you use to add these is the same kind of thing that we do for cysteine chemistry or lysine-like chemistry with the epsilolamines. So it's exactly analogous. And we typically get, on average, about 200 or so metal atoms per antibody. And then both Scott's lab, Scott Tanner, I know, and Mario are doing some work with nanodots or nanocrystals to be able to essentially replace these like a quantum dot with a large crystal that has obviously between 10 to 20,000 ions per antibody. We've also got some of those worked on as well, and they look like they're working pretty well for the surface proteins at least. So again, it's one cell per shot in at a time. you get about 1,000 cells per second on average, but maybe, I think working maybe 500 to 600 cells per second. And they're just literally shot one at a time through the flame. If you think about it as they're coming in, one individual cell, as it goes in, becomes a small plume of ions about a millimeter or so in circumference, right, or sorry, width. And then they're sucked in here. Right now, only about 30% of the cells are actually making it from here through to there, which you think about the size of this, relative, it's like basically shooting a BB across campus and getting it into Lee's teacup on the far side of campus, right? So the fact that they even get 40% of the ion sets to here is really a surprise, or is great. What we think is actually going on, I've been talking with Scott Tanner a lot about it, what we think is going on is that actually as the cells are being shot out into individual cell droplets, they're being fragmented. So, because when they've been able to get up to 70 or 80% of beads into the right place, right, they've been able to increase the specificity that way, but when you then take the same modifications that you've done and use that on cells, you don't get the same. So they're being chopped up, we think. And so that's what we published in Science in May of this year. So a little bit closer, and this is kind of, I think this is more for Dave Parks, fluidics and whatnot. It's plasma and gas fluidics. So the same idea, right? A sample and gas plus cells comes through the middle. There's an auxiliary gas which goes in on the outside. And this is the torch is out here. So there's actually these rings here are actually an electromagnetic coil that's pumping radio frequency into the argon, which is then heating it, right? And then you spark it, you light the thing, and so from this point on, you're getting increasing temperature, and at the center is where the plasma flame is hottest, where you're hoping to get complete ionization of the cells, atomization and ionization of the sample. And then there's a cooling gas on the outside here to help focus this. So luckily, a lot of the work that's gone on for the creation of this went on not because of Scott, but went on because of the semiconductor industry. That this same tool is used in semiconductors to ensure appropriate doping and then also ensure that there's not inappropriate metals or other contaminants. It's actually interestingly also used in metal and gem provenance. So if you want to know if the diamond came from Zaire or if the diamond came from Brazil, you would scrape a little bit where the person wouldn't notice from the edge of the diamond or the metal. You drop it into an ICP-MS and it will give you a signature. And there are presumably signatures of the mines and materials that come from those mines. So it's a well-developed field that Scott Tanner took advantage of. And this is just some more information about where the signal is coming from across the width of the flame. And then just one last version of that. So this is where the nebulizer is making the single cell droplets, and then that just goes in. And then from this point on, obviously, it's a gas, and then it's just a time of flight measurement, relatively straightforward, actually. And so obviously, we're not using lenses, we're not using optical lenses, we're using electromagnetic lenses to move the ions around by standard mass spectrometry approaches, right? And so when the ions come through, the carbon, the hydrogen, the oxygen are all basically shunted aside and thrown away, and then they're focused entirely on the mass range that we're measuring. So if you go to the worst of the ions or the isotopes versus the one that is best, at least across this class, it's only about a 3-fold difference in terms of the sensitivity, which is nice because we know the range of quantum efficiencies you get with the fluorophores, 10 to 50-fold. So that's nice. And detection is completely linear. So across, as I said before, up to 10 to the 8th, 10 to the 9th fold range. And then we did for the science paper, we had to do an awful lot of comparisons of what kinds of signals you get with mass cytometry versus fluorescence cytometry. And of course, we had to satisfy the reviewers. And one of the issues, of course, if you've been in flow cytometry for all these years, we expect to see populations, even populations that are zero, having nice fuzzy circles, right? Which of course, much of this fuzziness even down at the zero is due to autofluorescence. We don't have any of that. So at least in our initial paper, we didn't do too much modification of the numbers around here. We just let them sit around zero, which of course can give you some issues if you have to make divisions into zero. But that's nice for us because even though this device is maybe 5-fold less sensitive than some of the average fluorophores, where it matters most for low-level detection, you don't have to contend with autofluorescence. So signals can be nice down here. We've developed a couple of tricks for amplification now of the signals, so we can actually amplify with DNA chains, where with an approach like this, we can actually see down to 5 copies of a given molecule per cell with that kind of amplification trick. So we did, of course, all kinds of comparisons of what we see by mass cytometry versus what we see here with signaling proteins and surface proteins, and it, for most intents and purposes, is just what you want to see. So this enables us now to do some pretty incredible experiments. It enables us to not just look at surface molecules, but that would call out most of the major cell types, but we can look at signaling inside, right? And we've also built panels for cell cycle, DNA damage response, apoptosis, survival, metabolism. We're actually going to be building a cysteine system for measuring oxidation, redox as well. So this is a list of the molecules so far that we've validated for CyTOF. All the kinds of surface molecules you would want. You can actually measure cytokines as well, and we can measure RNA, as I mentioned before. And here's a lot of intracellular proteins that are in alphabetical order. But notice that a lot of these are aimed at phosphorylation sites, because that's kind of the bread and butter of a lot of the stuff that we do in the lab. But you can rearrange these, obviously, beyond alphabetical into things like, again, cell cycle, methylation. Anything that you can get an ion tagged onto is really fair game. So we go from this, when I was just a little newbie grad student, to this more or less when I left, still up to today. I mean, Mario would be out here, right? And then 32, this was at the time of the publication, but we're now doing 45 parameters in the lab now. So that's not a scalable solution for visualization. So I know that there's a lot of ways to do clustering and visualization. So we went back to one system, which was to how could we deal with 45-dimensional data in ways that seem intuitive. And so, you know, we're accustomed to thinking of the immune system as sitting in these blocks, and we forget about the arrows, right? The transitions. One cell becomes another cell, right? Markers on the cell surface or intracellularly go up and down in coordinated patterns. So what we decided to do was to see if we could use an algorithm that could reconstruct the progression on the basis of following a most likely path, right? So that, you know, if you were just a standard immunologist, if you saw 5 markers on a cell, you'd probably place it here. If you see a different set of markers, you'd place it there. All we're doing is we're saying we're going to take any one cell, and we're just going to put it in a line, and then we're going to begin to pick amongst all of the other cells to see which one is most like it, most like it, most like it, and build out a path. And that's what we did. So you all know that various markers go on and off during this. So computationally, to deal with that, what we did was we collected cells by looking in a clustering in n-dimensional space for regions of similarity. We call them metacells. We then connect the metacells with paths or nearest neighbors, right? So it's actually a minimum spanning tree is what we use. And then we use then that connectivity and most likely nearest neighbors to reach into the sort of, imagine a dimensional portal here, 40 dimensions of a complex point cloud. We reach in, grab any one of the metacells, pull them out along the string, and the string is actually two-dimensional. So now we can flatten the whole thing down into 2 dimensions and look at the whole immune system at once and never even think about all the dimensional information. So this is sort of a representation of how the system works. One marker goes down, another goes on, right? That's a progression. But we know that other markers come on and off all throughout the system, right? So that's the immune system, or any progression that you see in an experiment. But when we take, say, the bone marrow out, we no longer have the connections nor any of the order. So that whole business that I just told you about doing the realignment, actually a 10-year-old could probably do it, right? Take one cell, find the one that's most like it, et cetera. And if the 10-year-old's smart, they realize that there's branches, and you reorganize it. So part of this was in the science paper, and then the rest of it just came out in the Nature Biotech paper just last week. And so the program is written so that you have these structures. You can click on any of these individual little blobs here, and it brings up the underlying two-dimensional data, so you can go back and fact check and reality check on it. And we actually now have automated ways to do that. So that, for instance, is the whole immune system at a glance. That's the CD45 expression. And it organized the tree by most likely similarity, right? It's not a progeny tree. Don't mistake it for a progeny tree. It's a most likely similarity by surface markers tree. And then we color this according to the, say, CD45 or CD3, and then the CD8 sub-branch of the CD3s. You can, once you've captured them in these meta regions, you can then start to do all kinds of interesting math with them. And one example of the math would be to say, let's say we're looking at phosphostat 5, basal. So there's a couple of red blobs, interestingly, here that are high for those. Those are actually blast cells that are doing something with phosphostat 5. If you hit the cells with IL-7, the T cell and NK T cells turn on. But again, this notion of being able to capture them, I can divide the cell blobs in this into this, right? Remap the colors, and now basically everywhere where the blobs are black, nothing happened to phosphostat 5 in the presence of IL-7. But this tells me that all of this is activated, right? So that tells me where across the whole immune system it's activated. There's just a positive control for B cell receptor, right? PLC gamma is downstream. And then this is a nice illustration because the LPS receptor is CD14, so we stained for CD14, and you can see that it's nicely on in this region. But if we look at the activity of P38, a phospho-P38, a MAP kinase, downstream of LPS, which is binding to the CD14, which is a co-receptor, you can see that you can see signal here that you can't see there, right? You can't see CD14. So obviously we've gone below the level of detection of CD14, but we're able to use the amplification signal to read out the presumed presence of low-level CD14 here. And so finally, because I know people will want to either talk about other things or hear the next person.
It's very beautiful, Gary. It's really, really
We've
beautiful.
used this now to think about cancer, right? In that I think of cancer as a form of parallel pseudo-differentiation. Just because a cancer cell has mutated doesn't mean it doesn't still try to do what it wanted to do previously when it was a normal cell. So to what extent do they still follow the same program? Or at what point, once they've gone beyond normal programming, do they start to do their own sub-programming? So this is just one example of a pediatric ALL, right, a B-cell ALL. 95% of the bone marrow of this patient is tumor. The algorithm still nicely finds and organizes the rest of the immune system, the normal infiltrating immune system, and then here are the blasts. This is what a pathologist would call the disease. But interestingly, if you go in and look at what the markers are, color by biology as we call it these days in the lab, where the normal pro-B cells are, or at least cells that have markers of pro-B expression, look at how many of them there are relative to the rest of the cells. Obviously, these cells have grown outside of their normal bounds. So whatever the mutation is or the environment of the tumor is, it has caused those cells to grow out. But then what fascinated me was this complex structure in the leukemic blasts. So the only way that you get these kinds of branching structures is if you're getting the same pattern occurring again and again. So what that's implying is that there's some process that's happening again and again and again. It's recapitulating. Interestingly, if you ask where certain markers like Ki67, marker of cell activation and cell division, they're out at the periphery of the branches. And if you begin to trace back, you can see that, well, that's high, low, 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 back to the core. So presumably there's a stem cell-like cell down in here that's driving that. And then you can ask other kinds of questions about where are various proteins activatable across those trees. So interesting, there's the so-called heterogeneity of cancer. And then there's cells that are resistant to dasatinib inhibition. Right, so you got the core of the tree is inhibitable by dasatinib, but these cells are not. So are these then the resistant cells? And I won't go through this. Just to say that we've been able to do this now with solid tumors. This is now 41 primary ovarian cancers from women, serous. And the fascinating thing about this, I'm just going to sort of jump to the conclusion of this, is that all 41 patients, all 41 of these serous patients, basically when you build their tree, it shows that all 41 are following the same kind of pseudo-differentiation program, right? So that what it's saying to me, at least, is that whatever the mutations are, genetically dissimilar, obviously, women from the start, they're falling and following down one or more of the same paths. Otherwise, we wouldn't be able to find this tree. Now, what's fascinating, though, is that different women populate the tree differently, meaning that whatever the mutation is, so the stem cell is actually up here. This little cell up here, that's the stem cell of the tumor. So this woman, 3336, her tumor grows out the cells here, and then these grow, and then out here. This woman does not. The central piece of the branching structure is what this woman. Now, nicely, people recapitulate. So two different women, similar physiologies, but this woman has this piece of the tree. This whole set of the tree is filled with pluripotency markers, right? So if you look at the markers of pluripotency, NANOG, SOX, et cetera, this woman obviously has both a stem cell disease and a more mature disease, et cetera, et cetera. And so the next stage of this is to look at drug reactivity. All of these women would be considered the same class of disease. And yet, by virtue of surface markers, we know that each of these women has actually very different diseases. Some of these are immature, some of these are one kind of mature cell, or other kinds of mature cells. So no wonder most of the time standard chemotherapy doesn't work for these people, because they've just got extremely different diseases. So that's basically it. And I think I'll just stop there. There's tons more slides, but yeah, this is the Herzenberg lab. Everybody has tons of slides. So anyway, thanks a bunch. Kathy.
In your dasatinib-resistant cells, have you now gone back by FACS to sort those out, and can you actually find the typical mutation?
That's the next step. We haven't done that, but I mean, unfortunately, obviously, as Len likes to remind me again and again, the cells are dead, and so therefore it will not replace the sorter. So what we've done then are I don't say that, Len. I've never said that.
Oh,
I've
Gary,
never said
have you
that.
on tape at a cytometry meeting.
I said I was gonna,
Crazy.
I was going to put my LSR2 on eBay.
You said Craigslist. You said Craigslist.
Craigslist, okay, Craigslist.
But Gary, it is absolutely true that a lab interested in this stuff can find enough to do with this machine that it doesn't need a sorter for, depending on what you're looking at. And the things are complementary, I mean, you
Yeah.
haven't said so, but obviously, if you pre-sort the cells so that you can get enough of a concentration of the cells you're interested in, then you can do the number of cells within a reasonable amount of time on the machine.
Yeah.
If you try to do something that's a 1% subset, it's not going to work.
Yeah.
You can pre-sort them and then go with it.
Absolutely.
So the two machines are complementary. They're not really antagonistic, and they just shouldn't be seen that way.
I don't see them that way. I
So don't
mean,
send you put it on eBay, you can use it.
I know. It was like the same time that I said that I was going to check one of the fax machines with the second fax machine. Yes. Yes.
You know, I have to say, I just have to make a comment about that, because Dave Parks was just presenting different nanoparticles on different machines, and I was wondering if your experiment.
Vindicated at last. It took 20 years, but I'm vindicated. Well, but it's not as good as the time that Tim Gaddis called down from the Mouse House to say Gary Nolan wants to know whether he should take the left spleen or the right spleen. I was a plant biologist before I came to this lab, so.
Along those same lines, you know, Mario was just pointing out the use of the stain sets and stuff. One of the things that I was trying to tell Scott Tanner when he thought I was insulting him was that the use of cytogeny, where you've got 50 markers that you need to keep track of, is a phenomenal use of this. You don't have to use the stain set design thing to
Right.
use it, although even there it might be useful in different ways. But just listing these reagents and having them available when nobody can remember what the names of all these elements are
Right.
would be extremely useful. So let's talk about that a little further.
Yeah, that would be great. I mean, I think what's been nice is about the system itself. We all know that compensation is like the big boogeyman in the design, at least, of panels. And I've had basically first and second year grad students come in and they're doing 30 parameter experiments from the get-go. I mean, the machine.
They can analyze them when they say.
Well, they can't analyze, no, I mean, even Spade has its own problems in that, we generate bazillions of Spade maps, and then you still got to go through them, and so now our next step is, frankly, we're trying to figure out how to do regression analysis, directed regression analysis that will automate the findings for you to compare things.
What we've done with the cytogeny is get rid of that compensation problem so that second-year students can come in and use 12 color and really not have to deal with it.
Right.
And as far as we can tell, it does as well as the experts do, and we're now going to throw it out there and have the real experts try it in all their different ways. But this is the whole concept. It's really to get these things together in one way and another, and I would really like to just see this, go on to happen. We should throw compensation out
This is only analog here.
from ever being in anybody's stuff. What?
He's just,
Speaker?
He'll introduce himself because we don't have much time. But you're gonna love this, Fred.
You haven't remembered dinner, those who have been invited, Peking duck, don't know where it is, and come.
So Gary, the question I always ask you, and George's telling me, that
Wait a minute, Gary, pay attention.
I'm listening,
Anyone wants to ask
I'm
me a
listening, I'm just trying to help him get
question?
this.
Speak out loud.
I'm speaking out loud. So, sorted, I mean, you say that's coming, it's coming. Is it really?
Well, this one? So it's possible, is what I said. So that you can sort with this machine, I believe, if you separate the ions from the cells before the ions go into the machine. And so just like you did 30 years ago, just keep track of where things are. Now, I said you could do it, but it's paper. I mean, we submitted a patent for it. I have no intention to use it any more than just to, frankly, is to make Irv stop bugging me about it.
Did it work?
No.
Hey, Gary.
We found something else to bug me
Gary,
about.
we have to shut the video off. Oh, that's all right.
Okay.
No, it's okay. I thought John was saying we had to leave, but
We have to leave in a half an hour.
we have half an hour for discussion, so we have time. Adam is one of our youngest sort of join-uppers. He's going to be an assistant professor here at Stanford starting next September. But he just recently walked into the lab through a mutual connection, and it's just fascinating what he's doing. And I realize particularly it's fascinating for our interests in redox, which haven't really been discussed here very much. So I just asked him if he could present this piece of technology.
Hi. So I just prepared just a very, very few slides. I have literally 5 slides. I'll do it very quickly. I've actually done my PhD here. I was in electrical engineering, and just a few months ago moved to UC Berkeley for a postdoc in chemistry. So I'm in a Caroline Bertozzi lab working on glycobiology. But what I thought I'll briefly take you through today is just a really cool, maybe, approach that ever since I started to talk to Lee and Land, we thought that it'll be really interesting to try to see whether there's any interesting complementaries between the two. And this is actually an approach that was initially thought for in vivo imaging. So the idea of trying to go in a molecular imaging approach. I did my PhD here with Sam Gambier in the radiology department. And the idea was to try to think of a physical way, of a physical effect that would allow us to see very deep into an animal and get some molecular information from the tissues for the purpose of tumor imaging. And this is the approach. The concept is called photoacoustic molecular imaging. And photoacoustics, for those of you who are wondering, is actually a very interesting physical effect that was discovered over 100 years ago by Alexander Graham Bell, where light is actually inducing sound waves. And in this way, what you're actually having is, if you tune the parameters in the system correctly and use the right particles, in this case this is a carbon nanotube, you can actually make carbon nanotube absorb light pulses that are in the near infrared or visible or whatever wavelength that you care to use. And these carbon nanotubes would actually absorb the light, produce a little bit of heat, and this heat will cause the molecules just around the particle to expand. And this expansion would happen rapidly enough to create a pressure wave. And these pressure waves in the right frequency will actually turn out to be ultrasound waves. And so we have light pulses go in and ultrasound waves coming out. And the cool thing about it is that now if one is interested, for example, in trying to image a certain molecule in the body, you can take those carbon nanotubes, and there are multiple kind of chemistries that one can use to conjugate